Good afternoon to all the EAPCET aspirants. So here we are in our EAPCET PYQ practice of the chapter Respiration in Plants. In our previous class, we are dealt with photosynthesis in higher plants EAPCET PYQ exercise, and now we are going to start with respiration. Look at the first question, which is match the following type of question. So here respiratory intermediates are given which are oxidized, and here the compounds which are formed after oxidation are provided. This is completely from TCA cycle. The concept is taken from TCA cycle. Fine, right? Here we go with first succinic acid. Succinic acid after oxidation turns to what? Fumaric acid, which is option 4. Okay, and then alpha ketoglutaric acid, which is 5-carbon compound, after oxidation gives rise to succinyl coenzyme A, right ma? Next, isocitric acid, which is 6-carbon compound, isocitric acid. This gives rise to oxalosuccinic acid. Pyruvic acid, in the process of oxidative decarboxylation, which happens in mitochondrial matrix after glycolysis, where pyruvic acid gets oxidized and it forms 2-carbon acetyl CoA, which is 2-carbon. Fine, right? So here you see A is 4, B goes with 2, C 3 and D is 1. Option D will be the right answer. Clear, right? Any doubts anywhere, ma? Yeah, once look into the theory also. Succinic acid oxidation by succinate dehydrogenase will give rise to fumaric acid. Alpha ketoglutaric acid, which is 5-carbon compound, after oxidative decarboxylation, this gives succinyl coenzyme A. Isocitric acid, which gives rise to oxalosuccinic acid, which is intermediate during oxidation. Next, pyruvic acid will give rise to acetyl CoA in link reaction or transition reaction, which happens in mitochondrial matrix. Second question you look into, ma, this is on energetics of C3 cycle. To reduce 1 carbon dioxide in C3 cycle, the assimilatory power needed is, always do remember whenever they are asking you assimilatory power, which includes both ATP and NADPH. Fine, right? This requires 3 ATP, 2 in reduction, 2 ATP in reduction process, and 1 ATP is required for regenerating RUBP. 1 ATP is required for regeneration. Fine. And how many NADPH are required? 2 NADPH are required for the process of reduction. So here we go, how much ATP and NADPH required? 3 ATP and 2 NADPH. Clear with the answer, ma? Next question. This is again from TCA cycle. If you look into EAPCET PYQs, whole lot of questions are put on TCA cycle itself. Many questions are asked from Krebs cycle itself. That important is this phase of aerobic respiration. Look into here. In Krebs cycle, the reaction representing substrate level phosphorylation is. Substrate level phosphorylation is where succinyl CoA along with the reducing enzyme, this gives rise to succinic acid. Okay, where here this phosphorylates ADP or GDP gets phosphorylated from the succinyl CoA and this forms ATP. Clear, right? So the answer will be B. Fine. Any doubts anywhere? Clear till here, ma? Yeah. Substrate level phosphorylation occurs in this step where succinyl CoA phosphorylates ADP or GDP and this forms succinic acid. Look at the next question, which is on ETS. So, yes, this question is on ETS. Look at here. ATP formed by electron transport system in aerobic respiration from coenzymes formed in one Krebs cycle. Okay. In one Krebs cycle, how much amount of ATP is formed by ETS in aerobic respiration? Only from Krebs cycle. So, in one Krebs cycle, if you see, 3 NADH are released and 1 FADH2. Do not count down the ATP because it does not require any coenzyme to reduce. Okay. So, 3 NADH, 1 NADH is equal to 3 ATP in approximately. So, 3 into 3 is 9 ATP in total, and 1 FADH2, 1 into 2, that is 2 ATP. So, total it is 11 ATP. Fine, right? Option A will be the right answer here. Clear, ma? Any doubts anywhere? Yeah. Next. What is the ratio of total ATP molecules formed to net ATP molecules in oxidation of a glucose molecule, oxidation of a single glucose molecule through glycolytic pathway? Okay, ratio of ATP molecules to net ATP, total ATP molecules to net ATP molecules. First, you recall at which steps ATP is being utilized. ATP is being utilized at the first step and third step, where 1 ATP is utilized by the glycolytic pathway. At sixth step, 1 NADH, you can consider 2 NADH because it will be 2 G3P from single glucose. So, it is 2 NADH and at the seventh step 2 ATP is being released. At 10th step, where phosphoenol pyruvate is converted to pyruvate, which is the final product of glycolysis, again 2 ATP is released. So, the gross ATP released is 4 ATP. Total ATP, how you have to calculate, ma? 2 NADH when it gets oxidized to ATP, 2 into 3, it is 6 ATP plus 4 ATP, which is gross ATP that is released in the 10th and 7th step, 4 ATP. So, it is total 10 ATP, right? 10 ATP is the total ATP, and net ATP if you consider, okay, net ATP if you consider, net ATP will be 2 ATP, okay, 2 ATP. Fine, net ATP is 2 ATP because of 4 ATP that is being released, 2 ATP will be utilized at first step and third step. So, net ATP will be 2 ATP. If you have to consider only ATP excluding out NADH, then it will be 4 ATP, ma. Here the term total is used, but nowhere in the question he asked about NADH. So, let us go with the total ATP which is released in the 7th and 10th step only, which is 4 ATP. Fine, right? If NADH is also asked, then go with 6th step also, then total ATP will be 10 ATP. But in the question, only he has asked the ratio between ATP total which is released and net ATP that is being released. So, let us consider total ATP which is released at 7th and 10th step and net ATP after being utilized in first and third step, 2 ATP being utilized. So, the net ATP will be 2 ATP itself. So, here 4:2 will be 2:1, right ma? So, the answer will be A. Clear? Clear till here? Any doubt anywhere ma? Yeah. Enough. Look at assertion reason question. Complex phi in assertion. Complex phi consists of two major components. One is F1 and the other is F0. Two major components which is F1 and F0, which are useful in the production of ATP. This is true. F0 is peripheral membrane protein and F1 is integral membrane protein complex which forms a channel. Wrong. Once you recall it, if we consider this as the mitochondria and this is the inner mitochondrial membrane or cristae, let us consider. Fine, right? So, here from intermembrane space to mitochondrial matrix. Fine. So, here F0 F1 complex, if you consider, F0 is embedded inside the membrane in cristae and F1 is towards the mitochondrial matrix. Fine, right? And in matrix itself ATP is formed. Here if you see in reason, F0 is a peripheral membrane protein, he is saying. F0 is integral membrane which is forming the channel, and in the reason F1 is described as integral membrane protein complex, which is wrong. F1 is peripheral which is directed towards the matrix. So, here assertion is true and reason will be false. A is true and R is false, where option C will be the right answer. Clear ma? Next question. In which form are proteins and fats utilized into respiratory pathway? They are utilized into respiratory pathway. In which form are proteins and fats being utilized into the respiratory pathway? The question is being asked from amphibolic pathway chart. Look at here. Proteins after deamination, they form as amino acids, true. Fats into fatty acids and glycerol, correct. Fatty acids as acetyl-CoA, true. Glycerol is converted to phosphoglyceraldehyde. Dihydroxyacetone as glyceraldehyde 3-phosphate. So, here if you look into the statements properly, in which of the following, in which form are proteins and fats utilized into the respiratory pathway? How they are coming into the respiratory pathway is important. Fats oxidizing into fatty acids and glycerol is true, but they are not entering into the respiratory pathway. Next, glycerol is converted to phosphoglyceraldehyde is true, this step is true, but at this step they are not entering into the amphibolic pathway. Here, when DHAP is isomerized to glyceraldehyde 3-phosphate, at this step they enter into the respiratory pathway. So, here if you see A, C, E are the right statements. These are the steps where they are entering into the respiratory pathway. Amino acids, they will be directed as pyruvic acid in the amphibolic pathway. Fatty acids as acetyl-CoA and DHAP is isomerized to G3P. It is A, C, E. Option C will be the right answer. Clear ma? Next question. Assertion, reason, type again. Under anaerobic and aerobic conditions, lactic acid and pyruvic acid are formed respectively in skeletal muscles, whereas in yeast during fermentation ethanol is formed. True, right? Under anaerobic respiration, lactic acid is formed in the muscle cells. Okay, this is because of the immediate requirement of ATP at the time of exercise. And during aerobic conditions, pyruvic acid is formed. Fine, right? And they are formed respectively in skeletal muscles, where in yeast cells, after alcoholic fermentation, ethanol is formed. In reason, if you address metabolic pathways give variety of products based on the conditions. True. Metabolic pathways will give variety of products based on the conditions. So here if you see, based on conditions, whether it is aerobic or anaerobic, and based on cell. Assertion is true and reason is true, which explains A. Option A is the right answer over here. Fine. Next. Look at this. Arrange the following substances in sequence as formed in EMP pathway. EMP pathway is what? Glycolysis pathway. That is why I told you synonyms are very important. Due to transfer of a group. Okay, we have to arrange them in sequence. First it is glucose 6-phosphate, which gets turned to fructose 6-phosphate, and then this glucose 6-phosphate isomerizes to fructose 6-phosphate. Later fructose 6-phosphate phosphorylates to fructose 1,6-bisphosphate, and then phosphoenol pyruvate is the last step, followed by 3-phosphoglyceric acid, which is the 8th step. Fine. 3-phosphoglyceric acid to 2-phosphoglyceric acid, after which you will get phosphoenol pyruvate at the 9th step. Fine, right. So here transfer of a group, due to transfer of a group. Okay. So this will be the sequence of the reactions, but we have to address only those substrates which are formed due to transfer of the group. So if you see 6th step, phosphate group is transferred at 6th carbon of the glucose, which is forming glucose 6-phosphate. Clear, right? And then B, if you see fructose 6-phosphate, A, and then E. Okay. In phosphoenol pyruvate formation, there is no transfer of any group. Okay. So here we will go with option B. Clear, ma? And then choose the incorrect statement. Ubiquinons also receive reducing equivalents from complex 2. Yes, they do receive reducing equivalents also. Statement 1 is correct. In coupled reaction, GDP formation, ATP is synthesized. GDP formation, GTP will be synthesized, not ATP. If ADP is formed, ATP will be synthesized. The first member of TCA cycle is oxaloacetic acid, which is true. Pyruvic acid will be moved into the TCA cycle. Pyruvic acid is not moved into the TCA cycle, it is converted to acetyl-CoA. You can either choose B or D. Let's look at the explanation once. Yeah. Match the following type of question you see. This is glucose list 1 and list 2. Which one will enter the respiratory pathway, he is asking. Okay. Glucose gets phosphorylated to glucose 6 phosphate. So, A will be 2. Okay. Glycerol turns to phosphoglyceraldehyde, which is option 1. Acetaldehyde from pyruvate amino acid to acetyl CoA. Amino acid will enter the amphibolic pathway through pyruvic acid. Clear, right? Fatty acids after breakdown, they will enter to the amphibolic pathway through acetyl CoA. This will be the intermediate in the process. Clear? So, C is 4 and D will be 3. Glucose after phosphorylating, it turns to glucose 6 phosphate, glycerol to phosphoglyceraldehyde. Fatty acids will join the amphibolic pathway as acetyl CoA. C will be 4. Amino acid will join the amphibolic pathway as pyruvate. Clear? Next. Look at this question. Assertion reason type again. The pathway from glucose to lactic acid occurs in 10 metabolic steps. Organisms will trap the energy and they store it in the form of chemical molecules or chemical bonds. True. Glucose is the only substrate for the production of energy. No, there can be many respiratory substrates. It can be fats and proteins as well. Fine. So, here A will be true and R is false. Option C will be the right answer. Clear, ma? And then in ATPase enzyme, F0 part is embedded in the membrane and forms a transmembrane channel. True. This forms a transmembrane channel. F1 part protrudes on outer side of the membrane and proton gradient is transformed across the membrane. True. Proton gradient provides energy for conformational change in F0, not in F1. It is in F0. Conformational change in F1 synthesizes ATP. Okay. Once again go through second statement. F1 part protrudes on outer side of the membrane and proton gradient is formed across the membrane. Yeah. So, here the right answer will be A, B and D. Option C will be the right answer. Next question, multi-statement based again. Look at it carefully. Which reactions do not occur in TCA cycle? He is asking which do not occur in TCA cycle. Reactions of pyruvic acid with OAA form citric acid. Yes, this forms in TCA cycle. Formation of 2 carbon dioxide molecules occurs in TCA. Release of 4 NADH plus H plus. No. Release of 3 NADH plus H plus in one TCA cycle. Formation of 1 GTP. True. Yes, formation of 1 GTP or 1 ATP is formed. So here if you can specify it, option C, I can find only option C because GTP is formed in TCA cycle. Fine. Next. Which enzymes of TCA cycle are located within mitochondrial intermembrane space? What are the enzymes present in mitochondrial intermembrane space and prokaryotic cytosol? Prokaryotic cytosol respectively. What are those enzymes which are present? So here if you see lactate dehydrogenase, no, it is not present. Succinate dehydrogenase, just a second. Look at the question carefully first, analyze it for a minute and then we will go for the answer. Yeah, succinate dehydrogenase is present in mitochondrial membrane. This acts towards the inner mitochondrial membrane, whereas lactate dehydrogenase is present in the cytosol of prokaryotes. In the question he is asking what is the enzyme which is located in mitochondrial intermembrane space and prokaryotic cytosol. In prokaryotic cytosol it is lactate dehydrogenase, and mitochondrial intermembrane space it is succinate dehydrogenase. So B and A, it will be option B. Next. Yeah, look at this question. Assertion reason type again. First cells lived in atmosphere lacking oxygen. All organisms retain enzyme to oxidize glucose completely without oxygen. No, you cannot oxidize glucose completely without oxygen. Okay. Did you look into assertion and reason? Yeah. Now we will clearly analyze the answer also. Yeah. Early earth was anaerobic, so assertion is true. Okay. First cells lived in the atmosphere lacking oxygen. Anaerobic species, especially archaebacteria, is the ancestors of prokaryotes. They are anaerobic, so assertion is true, and reason will be false because complete oxidation without oxygen is impossible. Incomplete oxidation takes place in the absence of oxygen. Next question. Identify substrate level phosphorylation reactions from here. Carefully look into it and analyze. SLPs. Done? Look at the answer. ATP is formed in glycolysis, GTP is formed in TCA cycle. So here if you see from triose bisphosphate to triose phosphate at the 7th step of glycolysis, which is the 7th step of glycolysis, okay, ATP is being formed and this is the substrate level phosphorylation. And here if you see from succinyl CoA to succinic acid, which is 4 carbon, succinyl CoA to succinic acid. Here again ATP is being formed, which is phosphorylated, okay, from substrate itself, that is succinyl CoA. So here also one more SLP is being formed in TCA cycle, okay, this is in TCA cycle. Option A and C will be correct here. Next. Biomolecule common to respiration of fats, carbohydrates and proteins. Which is the common respiratory intermediate for all the three respiratory substrates? That is being asked in the question. That is acetyl-CoA. Okay, because fatty acids will join the cycle as acetyl-CoA, glycerol will join as DHAP. Here I will tell you, see. When fats break to fatty acids and glycerol, fatty acids will enter the cycle through acetyl-CoA, glycerol through DHAP, or you can also consider phosphoglyceraldehyde. Next is proteins. Get deaminated to amino acids. These amino acids enter the respiratory pathway through acetyl-CoA. Fine, right now. So here the common respiratory intermediate will be acetyl-CoA. Later DHAP will continue the cycle, will continue the glycolytic pathway, and pyruvate converts to acetyl-CoA. Here also amino acids, they will enter the cycle as pyruvate, sorry. Pyruvate later gets converted to acetyl-CoA. Fine. Next. So the answer will be D. Match the following type again. Analyze the question carefully. List 1 and list 2 are provided. In list 2, enzymes are being given. Substrates and the catalyzing enzyme. Phosphoenol pyruvate is the substrate which gives out the product, that is pyruvate, by the enzyme enolase, which is option 2. Okay. Pyruvic acid is formed by pyruvate kinase, which is option 3. Triose phosphate is formed by aldolase at the cleavage step. Glucose 6-phosphate is formed by the enzyme hexokinase. Clear, right? So A will be 2, B 3, C 4 and D 1. Fine. Next. Look at this question. Identify wrong statement related to glycolysis. Glycolysis has 10 chain reactions. True. In glycolysis, ATP is utilized in one step only. No, ATP is utilized in 2 steps. First and third steps of glycolysis. There is one step related to the formation of NADH plus H plus. Yes, at 6th step, where glyceraldehyde 3-phosphate is turned to 1,3-bisphosphoglyceric acid. In D, glycolysis can take place in the mitochondrial matrix, also wrong. Glycolysis occurs both in aerobic respiration and in anaerobic respiration. This takes place only in the cytoplasm or in the cytosol, you can say. So here A and C, 2 statements are correct, but here in the question he asked this wrong statement. Whenever statement based questions are given in your exam, just look at the question carefully whether he is asking you to identify correct statements or incorrect statements. As he asked wrong statements from here, the wrong statements will be B and D. Option B will be the right answer. Clear, right, ma? And then identify the condensation and cleavage reactions in TCA cycle. Condensation and cleavage reactions. OAA and acetyl-CoA. OAA is 4-carbon compound, acetyl-CoA is 2-carbon, which forms citric acid and coenzyme A. True. Fumaric acid, which is 4-carbon compound, is turned to malic acid, which is the last step of glycolysis. Here he is asking us to identify only the correct statements. One is for condensation. First step of TCA cycle is condensation. And next is cleavage. This is not cleavage step. Cis iconitase to isocitrate is also not cleavage. Succinyl CoA to succinic acid. Yes, CoA is being removed from succinyl CoA, where succinic acid is formed after substrate level phosphorylation. This will be an example for cleavage. So, here A and D, option C will be the right answer. A and D, fine. Next, match the following type of question again. Complex 1 is NADH dehydrogenase, which is option 5. Complex 2 is succinate dehydrogenase, which is option 4. Complex 3 is cytochrome BC complex, which is option 2. Complex 4 is cytochrome C oxidase, which is option 1. Complex 5 is ATP synthase, which is option 3. Fine, right? So, A5, B4, C2, D1 and E3. Clear? This will be the correct match. Last question of today's session. Last question. Find out in which enzymatic reactions NADH is converted to NAD plus. Look at it carefully, all the steps. Acetaldehyde to ethanol. This is alcoholic fermentation. So, in alcoholic fermentation, let us observe once option A, right, which is alcoholic fermentation. In alcoholic fermentation, it utilizes the NADH which is released at the 6th step. Fine, right? At the 6th step of glycolysis, whichever NADH is released, that is being utilized by acetaldehyde to get converted to ethanol. So, here when acetaldehyde is converting to ethanol, NADH is oxidized to NAD plus plus H plus. This NAD plus will act as a reducing equivalent, okay, for glycolysis again. As this is released into the cytoplasm, this will act as reducing equivalent for glycolysis again. So, option A will be right, okay, where NADH is converted to NAD plus. Glyceraldehyde 3-phosphate to 3-phosphoglyceric acid, okay. Here nowhere NADH is converting from G3P to 3-phosphoglyceric acid. Next, pyruvic acid to lactic acid again, yes, right. Pyruvic acid to lactic acid also, which is anaerobic respiration. Here by the enzyme lactate dehydrogenase, where NADH is converted to NAD plus, and NAD plus serves as reducing equivalent for the process of glycolysis. OAA to alpha ketoglutarate, no. Alpha ketoglutarate to succinyl CoA, no. At this step also there is NADH is not converted, rather NADH is synthesized in these two steps in TCA cycle, where this gives out the NADH. This gives out NADH in TCA cycle. So, here what is the enzymatic reactions where NAD plus is formed from NADH? A and C, fine, right. One is in alcoholic fermentation, second is lactic acid fermentation, which is option D. Clear till here, ma? Fine, right. So, with this we have exercised the complete set of EAPCET previous year questions, and in our next class we will look into plant growth and development EAPCET PYQs. If you are having any doubt from the questions that we have solved today, you can raise your doubts in the comment box, where I will address all your doubts, okay.